My topic I will be discussing is atherosclerosis. Now, initially, I thought arteriosclerosis and atherosclerosis were the same thing. Before entering Loma Linda University and taking pathology and cardiology, I assumed that atherosclerosis was thickening of the arteries due to an accumulation of plaque buildup. I assumed burgers, pizzas, greasy foods, and foods high in fat were the cause of the problem, and that it began as soon as you ate it. I thought that obese individuals were those affected by this disease, and that men and women could be affected equally. Now, after properly being informed on atherosclerosis, I've learned that there is more to it than a buildup of plaque and what the buildup is made of. Atherosclerosis can begin at an epigenetic gene transcription level and can cause an altered degree of cytosine methylation on DNA. This will recruit or block proteins that activate transcription. Now, atherosclerosis is a condition where lipid plaques form on the inside of the walls of the arteries. The development of atherosclerosis begins in a pro-inflammatory environment. TNF-alpha, which is a cell signaling protein cytokine involved in systemic inflammation, binds to the TNF-alpha receptor on the endothelium. This initiates NF-κB, a pro-inflammatory transcription factor, that will bind to a promoter that will cause monocyte to bind to the endothelium. Now, VCAM1, a vascular cell adhesion molecule, moves to the endothelium and binds to a monocyte. The VCAM1 gene contains 6 or 7 immunoglobulins domain and is expressed on both large and small blood vessels only after the endothelial cells are stimulated by cytokines. The monocyte will then enter the endothelium and turn into a macrophage. This will increase scavenger receptors that have an attraction for oxidized LDL that are at a high level due to oxidized stress of the pro-inflammatory environment. Now, these oxidized LDLs are taken by the macrophages that engulfs them and turns them into a foam cell. These foam cells are fat-laden M2 macrophages that serve as a hallmark of atherosclerosis, which is commonly associated with increased risk of heart attack and stroke as a result of arterial narrowing and hardening. Foam cells form the fatty streaks of the plaque of atheroma and the tunica intima of arteries. Cytokines and growth factors can promote the migration of vascular smooth muscle cell through the lamina, forming a cap that covers the atherosclerotic plaque. This results in the narrowing of the vessels and could potentially rupture, causing blood clots called thrombus. If the clot breaks loose, it could be known as an embolus. Once blood flow to the heart is reduced, known as a stenosis, or blocked by thrombotic occlusion, this can lead to a serious cardiovascular event. The second hallmark of atherosclerosis is due to hyperinsulinemia, which will result in smooth muscle cell proliferation and differentiation of synthetic phenotypes. This will lead to smooth muscle cells, the inability to relax and vasodilate, which will make the tissues tighter, causing a non-responsive, thicker, and less compliant blood vessel. Due to this thicker and less compliant vessel, it will affect the pulse wave velocity and how the vessel should be able to push that blood forward, maintaining the forward propagation that will lead to good perfusion. In addition, there is another cause of atherosclerosis, which is due to a prothrombotic environment. It is when the monocyte binds to the endothelium and migrates inside, causing engulfing of oxidized LDL, like I said previously, which will turn into foam cells. These macrophages will also cause MMP, which is a metal proteinase, and TF, a tissue factor, to be released, which will cause a prothrombotic environment. It will do so by degrading collagen and aiding in the coagulation pathway, cleaving prothrombin to thrombin, which will form a thrombus or blood clot. According to the old theories of atherosclerosis, hypercholesterolemia is a condition characterized by very high levels of cholesterol in the blood. Cholesterol is a waxy, fat-like substance that is produced in the body and obtained from foods that come from animals, particularly egg yolks, meat, poultry, fish, and dairy products. The body needs this substance to build cell membranes, make certain hormones, and produce compounds that aid in fat digestion. Too much cholesterol, however, increases a person's risk of developing heart disease. It was said that people with hypercholesterolemia have a high risk of developing a form of heart disease called coronary artery disease. This condition occurs when excess cholesterol in the bloodstream is deposited in the walls of the blood vessels, particularly in the arteries that supply blood to the heart. The abnormal buildup of cholesterol forms clumps, or plaque, that narrow and harden artery walls. As the clumps get bigger, they can clog the arteries and restrict the flow of blood to the heart. The buildup of plaque in coronary arteries causes a form of chest pain called angina and greatly increases a person's risk of having a heart attack. Also, there was an old theory called dyslipidemia, which is an abnormal amount of lipids, like triglycerides, cholesterol, and fat phospholipids in the blood. In developed countries, most dyslipidemias are hyperlipidemias, which is an elevation of lipids in the blood. This is often due to diet and lifestyle. Prolonged elevation of insulin levels can also lead to dyslipidemia. Now, dyslipidemia stands out prominently as it enhances the deposition of the fats in the arteries, thereby narrowing the lumen, obstructing the blood supply, leading to thrombolytic episodes such as stroke, transient ischemic attack, ischemic heart disease, and pulmonary embolism. Of course, this is now something that isn't the primary cause of atherosclerosis. Like I mentioned before, there are many causes of atherosclerosis. The main causes are due to the development of those foam cells that can develop in the plaque you see in the blood vessels. The prothrombotic state that can cause a thrombus, and the synthetic smooth muscle cells that will proliferate and differentiate, resulting in the inability to relax and vasodilate, which will make the tissues tighter, causing a non-responsive, thicker, and even less compliant blood vessel. There is also even the mitochondrial dysfunction caused by hyperglycemia that will cause an oxidative and pro-inflammatory pathways, which is shown on this picture. In addition, if the liver, the main organ for metabolizing, is experiencing dysfunction, it's going to increase secretion of prothrombotic inflammation markers, high triglycerides, and low HDL, which increase glucose uptake in adipocytes and increase free fatty acids, which can result in metabolic syndrome. This liver disease can cause atherosclerosis. Now, the most interesting mechanism to me was the oxidative stress and mitochondrial function. Mitochondria are the cellular powerhouses fueling metabolic processes through their generation of ATP. However, we now recognize that these organelles also have pivotal roles in producing reactive oxygen species and in regulating cell death, inflammation, and metabolism. Mitochondrial dysfunction therefore leads to oxidative stress, cell death, metabolic dysfunction, and inflammation, which can also promote atherosclerosis. The mitochondrial DNA damage is present and promotes atherosclerosis through mitochondrial dysfunction. Hyperglycemia is getting into the cell and causing the glucose overload in the mitochondria. When it works harder, it will produce more radical oxygen species and will do so a variety of things, such as a break in DNA, shown in this picture. It will inhibit protective mechanisms like eNOS and other pathways of oxidative stress like H. It will activate NFKB transcription, which will increase thrombotic environment genes. It will also increase cell proliferation and differentiation, and also it will increase monocyte bonding, which was what we talked about in the first slide. The high glucose load can also cause damage, tissue damage, because it's working the mitochondria too much, which will cause an oxidative and pro-inflammatory pathway. Now, this topic was interesting to me because before this class, I assumed that the mitochondria's function was solely on energy and forming ATP. I didn't realize the importance of this organelle and its role in atherosclerosis. Its function is truly a necessity for homeostasis, and when that balance is no longer being stable, such as in a disease or possible damage like hyperglycemia, it will cause these reactions, like an inflammatory state. Now, this inflammatory state will allow for the formation of foam cells to develop. It will also cause a reaction like NFKB, which will develop a prothrombotic environment that could lead to the development of a thrombus. In addition, it can lead to monocyte bonding and that oxidative stress that will create foam cells. In addition, the activation of a PKC pathway has been associated with changes in blood flow, basement membrane thickening, extracellular matrix expansion, increases in vascular permeability, abnormal angiogenesis, excessive apoptosis, and changes in enzymatic activity alterations, such as the sodium protein ATPase. The formation of the advanced glycation end products, or AGE, by non-enzymatic reactions is implicated in aging. The AGE products are involved in age-related pathologies such as atherosclerosis and neurodegenerative disorders, and in the development of long-term complications of diabetes. The neurodegenerative diseases often occur due to a loss of function or death of neural or neurological cells. Chronic inflammation triggered by intrinsic factors and nutrients affects tissue environment, especially in age-related diseases such as diabetes, atherosclerosis, and even cancer. So this mitochondrial dysfunction does a lot to our body and has a vital role in the development of atherosclerosis. Now that there's two foods that activate a pathway that inhibits or even reverses the atherosclerotic promoting pathway is garlic and pomegranates. Garlic has the ability to lower blood pressure, cholesterol, and blood clotting, has been recognized for some time and has been shown again in a recent trial. In addition, garlic, along with onions, provides a source of sulfur in the diet, which may be crucial for optimal amounts of antioxidants. In a study published earlier this year using baseline and follow-up CT angiograms of heart arteries, garlic reduces areas of low attenuation plaque at the 1-year follow-up. And pomegranates have powerful antioxidant properties and may improve the function of HDL cholesterol to boost the reversal of cholesterol transport that may reverse CAD. In studies of mice, pomegranates can reduce atherosclerosis. In humans with increased oxidative stress at risk for CAD, pomegranates reduced evidence of arterial damage. In another study using pomegranate juice for 3 years, the degree of narrowing in carotid arteries of 5 studies subjects was even reduced. Atherosclerosis is enhanced in arterial segments exposed to disturbed flow. The perturbed shear stress increases the expression of oxidative sensitive responsive genes in the endothelium. Now, evidence suggests that the polyphenolic antioxidants contained in the juice from the pomegranate can contribute to the reduction of the oxidative stress and atherogenesis. In one study that evaluated the effects of intervention with pomegranate juice and oxidative stress genes and endothelial enosynthase, or eNOS, expression induced by high shear stress in vitro and in vivo. Cultured human coronary artery endothelial cells exposed to high stress in vitro and hypercholesterolemic mice were used in this study. Pomegranate juice concentrate reduced the activation of redox-sensitive genes and increased eNOS expression, which was decreased by perturbed shear stress. In the cultured and in atherosclerosis-prone areas of the hypercholesterolemic mice. Moreover, oral administration of pomegranate juice to hypercholesteremic mice in various stages of disease reduced significantly the progression of atherosclerosis.